Hello guys, this video is about chemiosmotic hypothesis. So in the NCERT, in below the topic chemiosmotic hypothesis, NCERT discusses this, which is not actually chemiosmotic hypothesis. So we're going to clear that out in this video. Um, as we all know, this is the theme of right reaction. You see there is an electron transport system here. This is actually what is happening here. Whereas the chemiosmotic hypothesis is only what happens here. So I'll clear that during the course. But just because NCERT discusses this, I'm going to first talk about that. So basically in this scheme we see photosystem 2, the light harvesting complex, it accepts light, the electron goes to the electron acceptor, then you have the electron transport system. So this electron transport system is being discussed in detail here. So this first electron acceptor that we're talking about, that is pheophytin, which is what NCERT mentions is the carrier on the outer side of the membrane is pheophytin, not mentioned in the NCERT, but this is mentioned in the NCERT. The first oxidation of water happens where an electron is released. It goes to pheophytin, first electron acceptor, and then it is passed on to not an electron carrier but a hydrogen carrier, plastoquinone. Here I'd like to mention that in the ETS, that is electron transport system of the mitochondrial membrane, we have ubiquinone, which acts as the hydrogen carrier, and here we have plastoquinone, which acts as the hydrogen carrier. So this not only transports the electron to the next electron carrier, but also transfers a proton from the cytoplasm to the lumen or the thylakoid. So this cytosine B6F is the electron carrier on the inner side of the membrane. So we did pheophytin, we did plastoquinone, which is the H plus carrier, and then cytochrome B6F, which is the electron carrier on the inner side of the membrane. So then lastly comes the plastocyanin, which transfers the electron to the PS1, finally to the LHC of the PS1. And then the reaction that occurs is very well understood. This is what is the really confusing part. But all of this is not included in chemiosmotic hypothesis. What exactly is this hypothesis? I'll read out a line from NCERT. It is ATP synthesis. The actual hypothesis is that the ATP synthesis is linked to the development of a proton gradient across a membrane. So that is a hypothesis. It was given by, I think, Mitchell. So what does that mean? It's just that the proton pump or whatever various ways a proton gradient is being produced inside of the lumen where you have excess H plus here and less amount of H plus here. This is the electrochemical gradient. That gradient is used by the ATP synthase or the ATPase to do ATP synthesis. That is the hypothesis. Now, how exactly does that happen? So, if we see a detailed image of this ATP synthase, it looks like this. You have the F naught and the F1 units. The F naught has C units. This has gamma, alpha, beta, not required for NEET. But the only thing that happens is when an H plus crosses the membrane, when it diffuses out directly in the direction, the ATP synthase, what does it do? It allows the diffusion of H plus across the membrane, which releases energy to activate an enzyme that catalyzes the formation of ATP. But going into the detail, the breakdown of the gradient that provides energy that causes a conformational change. That means these units, they rotate. That causes the gamma unit to rotate, which causes the production of ATP from ADP and inorganic phosphate. So that is what ATP synthase does. The breakdown of the gradient provides enough energy to cause a conformational change or rotatory change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. To give you one point which is out of NCERT, with one rotation there are 3 ATP that are produced. So you can also say several molecules as mentioned in the NCERT. So that is all for this video. I'm not going to go much more detail. The only point that is Very important to remember that this is not chemiosmosis. Here we see the chemiosmosis, mostly the essence of the hypothesis that ATP synthesis is linked to the proton gradient, to the development of the proton gradient. And in this case, this whole cycle or scheme is helping in the proton gradient, whereas in mitochondrial ETS, we see the mitochondrial ETS helps in the proton gradient. So that's a different thing. So that's all for this video. I hope you learned something new from this. I'll see you next time. Bye.